Now, can you see now?
Yes, sir, it's visible.
Okay, okay, okay. Yeah.
Okay, sir, so with your permission, shall we start?
Yes, please. Yes.
A very warm welcome to all the participants. A very good morning. You are welcome to the webinar entitled Hacking God's Own Program: How Synthetic Biology May Change the Future of Engineering and Technology. It is organized by the Department of Basic Science and Humanities, Regent Education and Research Foundation. We are honored to have Dr. Sangram Bagh amongst us. He is an associate professor at Saha Institute of Nuclear Physics in the Biophysics and Structural Genomics Division. He received his doctoral degree from the University of Toronto in the year 2010. He was a postdoctoral fellow in the Department of Chemical Engineering, Massachusetts Institute of Technology from 2011 to 2013. He joined SINP in the year 2014, prior to which he was assistant professor at the Presidency University. He has been DST Ramanujan Fellow from 2015 to 2020. His research interests include cellular robotics and cancer gene therapy, space synthetic biology, quantitative reconstruction of native gene network. He has received numerous awards and has authored a number of publications in journals of international repute. We are very happy to welcome you, sir.
Thank you
Now
very much.
I would like Now I would like our honorable principal, Dr. Mahua Das, to address the audience. Madam, over to you.
Hello? Hello?
Yes, ma'am, you're audible. Yes,
Am
ma'am,
I
you're
audible?
audible. Yes,
Yeah.
ma'am.
Thank you, Anindita. Uh,
Thank you, ma'am.
Hello, everyone. Good morning to you all. Today, the Basic Science and Humanities Department has arranged the webinar on Hacking God's Own Program: How Synthetic Biology May Change the Future of the Engineering and Technology. I must appreciate the team for selecting such a relevant topic in the huge crisis through which we all are passing. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. Synthetic biologists or companies around the world are harnessing the power of nature to solve problems in medicine, manufacturing, and agriculture. It can be applied to help provide diagnostic tests and to develop diagnostic kits that are easy to distribute. It can also help us to better understand the COVID strains and create vaccines. However, I will not take much time because we have Dr. Sangram Bagh as the resource person for this webinar to get many important insights on the topic. I warmly welcome Dr. Sangram Bagh in our virtual platform. I also welcome all the participants in this webinar. I know that the session will be enriching as well as enlightening. The audience will have an enriching experience from this webinar. We hope that we can host Dr. Bagh at our Regent campus for more and more such engaging sessions after the pandemic is over. I take the privilege to thank Dr. Bagh for joining us and sharing his valuable time. Over to Anindita.
Thank you, ma'am. Thank you very much. I would now request our honorable Dean Academics, Dr. Ashok Kumar Shaw, to say a few words. Sir, are you there? Okay, maybe sir is not with us at this moment. Sir, Sir Sangram, sir,
Yes.
Dr. Bagh, then it will be over to you to take the stage and enlighten us with your speech.
Okay. And thank you very much. I'd like to first thank Professor Rajdeep Chakraborty and Madam Principal for inviting me in this session. And it's really an honor. And hope, as Madam Principal said, when this every COVID things is over, definitely I'll visit your campus. And thank you for the invitation, ma'am. So what I'm going to talk about today is a very, I do understand this is an engineering college, and I'd like to take as much as general, but the last few minutes, like 5 to 10 minutes, I would also like to talk about the what kind of work also we are doing in our lab. So the question is, when you all know that at this current moment, like, if the previous of the century people talked about is the century of the physics, and this century people tell that this is a century of the biology. And what does it mean? Because we can see that the biology is in a very complex system. So, but if you can understand a very little about that biology, we can change our future engineering and technology in many different ways. And you can see within the next 50 years or so, we go actually from whatever we have economy we have, we'll go towards something called the bioeconomy. It is starting from the chemicals, starting from the fuels, starting from materials, even the whatever your semiconductor chips and everything, which will be there would be a hand of the biology in that. And we'll see how that is possible by something called the synthetic biology in that. Now let us start from here. You probably actually see this kind of movie like the Jurassic Park. Either you have seen the Jurassic Park 1, 2, 3, or Jurassic World or something. It doesn't matter. But the whole thing is that this is a science fiction movie, and this is the first written in 1986, the Jurassic Park 1, what we called in terms of the movie, and it's written by Michael Crichton. The question is that was written in 1986. We'll just at this moment try to see that if we have the enough technology to make a dinosaur or not. So as you have studied, all of you, at least the biology in your high school, so you do know that either it's a dinosaur or a human being. The main theme or the what is the common among them is that the fourth thing, ATGC, 4 different nucleic acids, and that makes the genome either of a dinosaur or either of a human being. So if you have a dinosaur DNA, the first thing you would like to make that, can you read that the whole blueprint or the whole genome of the dinosaur? The question is that technology here? And it is. Probably you heard about the Human Genome Project, so which was like published in 2003, the first draft of the human genome. So, and you can see this is an industrial scale operation, and there's several countries, US and many European countries and Japan actually included, thousands of the scientists and $3 billion actually used here. And you can see its success has been announced by Bill Clinton, that time the president of United States of America. And I'll come why this is happening. A scientific project is basically announcing by a politician. So the whole thing, what the significance of this Human Genome Project is that we, at this moment, if we think the genome is the blueprint of the life or the software of the life, we can actually read that blueprint of the life by later by later. Okay, so that's the first part to make the dinosaur. Now, next I'll come that we can read. Now, can you write? What does that mean? So in 2008, the Craig Venters group actually, what they have done, they actually chemically synthesized the whole genome of a bacteria called Mycoplasma genitalium. Now, what is the significance of that? So you may heard there's something called polymerase chain reaction, which is, or PCR, which is basically amplifying a DNA template in millions or the billions of the copies. But that PCR is not writing that because it's like a photocopying. You must need a template in order to copy that. Whereas this chemical synthesis gives you the whole idea, you can write anything. So you showed that chemical synthesis of mycoplasma genitalium, the whole thing, but you can put any other chromosome in that way. So the significance of this artificial life, which I call artificial life 1, that you can write anything you want. And remember that this genetic substance is not biologically created. It is just created by the chemistry outside a biological cell, and you actually make this the whole genome. So this is called the artificial life 1. Now, what about in just after 2 years, 2010, the same group actually published in another important paper, and what I called it at artificial life 2, which is transforming one species to another, and you start from the scratch. What does it mean? They actually synthesize that whole mycoplasma chromosome and then put that chromosome in another species, and what they saw, this another species actually transformed to that the previous species. So this is like, if you want to see what is the significance in an engineering term, it's like software is creating its own hardware. What does that mean? Because I said that the blueprint of the life, or whatever the program is written, and that is the DNA, which has been synthesized chemically and put it in another species. So this another species changed and physically to another species. Another species means it's all proteins, all metabolites, all the cell wall composition has to be changed. It means that's the hardware to go, that's the life. And so the significance is software is creating its own hardware. But if you want to look this as in a machine world, how it looks like? It looks like this. You have heard or you have seen this movie Matrix. If you have not, I strongly recommend, but please, as an engineering student, you should watch this movie. And now here, if you remember this movie, or even if you don't remember, doesn't matter. So this guy is not a person, but this is according to the movie, he's an architect. It is basically a software itself. So this software actually developed all other softwares or all other programs in the Matrix. But that is software is creating its own software. But at the very same time, in this movie, this machine world, this programming world, they also create hardware. So the hardware machine, which are actually descend to destroy the human settlement at the Zion, according to this movie. So what I can say in terms of this in machine world, which actually looks like basically a science fiction movie, in biological world, the system is actually doing this for billions of the years by the evolution. So till now, what I got here, that we can read, and definitely we can write, we can transform to another species. So if you remember the Jurassic Park movie, there is something called the DNA video, which they actually show in the 3 steps they made the dinosaur. In the first step, they actually got the dinosaur DNA from the blood, which is like preserved for millions of the years under an amber. And from that blood, they got the dinosaur DNA. They actually sequenced this whole genome of this dinosaur. Then they actually chemically synthesized that whole genome and then put in a frog embryo. And this frog embryo actually changed with the dinosaur, right? So now, but I told you that by 2010, we had all things are in our hand. So we can read the whole genome sequence, we can write the whole genome sequence, and we can change that one species to another by putting this synthesized chromosome to the another species. So now, what I can tell that we are ready to make the dinosaur if you really get the blood of the dinosaur. Now, let me ask you another question. So, when you call about the engineering, whatever you're studying, your engineering, either electrical engineering, civil engineering, mechanical, whatever it is, so you always see that engineering like this, right? This is a beautiful building in Singapore, or this is a rover on the Mars. But I can assure you, you've never thought engineering like this, like a Pegasus or a Hawk. So if I ask a very fundamental question at this technological era, when you can actually control the movement of the rover on the Mars from the Earth with almost a mathematical precision, why we cannot make a Pegasus or Hawk? So what prevents us engineering a system like this? So in one way, the answer is very simple. And that simple answer comes from, if you want to make this building or the rover, you have a very well-established engineering design principle. And based on that principle, you first actually You actually fabricate it, then you test it. If there's a discrepancy, then you again come to the design, change the design, and you build it. But when it comes to the life, we do not know what is the design principle of the life, and that is the that hinders us to making or engineering system, something like that. Now, so in other words, what I can say that we can read the blueprint of the life, we can write that blueprint of the life. The question is, do we understand? For example, here you can see this is an blueprint of an engineering drawing, right? So now everything is written in English. Okay, all these geometrical shapes we do know, so we can read it, we can write it, but unless as a proper engineer you cannot get it what it has been written, so we don't understand. This is the same for the biology. We do not understand the biology at all in terms of its design. Now, but question is why you don't do that? So we have to go a little bit of history, and this guy is a Max Delbruck, who actually got the Nobel Prize in Physiology and Medicine in 1969 by discovering the replication process in a virus. Now, Max Delbruck was a very much actually is a gifted physicist. He was a student of Niels Bohr. Now you can imagine that time, that was the century of the physics, and everyone was so excited about the physics because in one hand there is like quantum mechanics, and another hand there is a theory of relativity. So everybody is so excited that they can find a new rules of the physics, because that time people started finding a lot of the new rules of the physics, and Max was not definitely an exception. So you'd like to get a new laws of the physics, but what he chose to look at the biological systems, because he believed that everyone is looking at the physical or chemical system to find the new laws of the physics. Why not look at the biology? We know very little about the biology, so I might get a new laws of the physics from the biology. Interestingly, when it comes to the biology, he found that he could not find any new laws of the physics from the biology, but what he found that biology actually follows the rules of the physics and the chemistry. And so he actually brought, he used to call is the father of the molecular biology, because as a physicist, he would like to see the biology from a molecular perspective. Now, we don't know probably, because whatever we are studying, either you are studying a basic science or either you're studying an like engineering, either you're studying a biology or the physics. So we all have somehow is drawn in the culture of that particular subject. And what is the culture of the physics that time, or philosophy of this physics, was a reductionist approach. So as a physicist, when he came to the biology, without his, he didn't know, but what he brought to the biology is that reductionist approach, and that is this. So what is the reductionist approach? So in the high school, when you actually solve the Newton mechanics problem, okay, so either it's an earth or it could be a car, it could be a ball, you always bring everything down to a point mass. Then you actually calculate the Hamiltonian of it, and then whatever you got the results, you mapped in that either it's earth or it's a car or it's a ball. So that is the particular reductionist approach of the physics. You bring everything down a simplistic way. Now that gives the biology a boost, because you know the reductionist approach in the biology gives a different philosophy, because you know that when people look for a particular gene responsible for a disease or a particular protein responsible for disease, they always actually brings this reductionist approach in the mind. And what is the highest manifestation of this reductionist approach is definitely this is the Human Genome Project, because this took like $3 billion of the money, and this money, most of them came from the definitely the government of United States of America. So to convince the politicians, the Watson had to give a lecture in US Senate, and suggesting that as the disease are connected with the genes, so if you know each of the genes of the human body, then we can actually cure every disease, including the cancer. But today, what we do know that from this Human Genome Project, whatever the knowledge we gathered, we cannot cure cancer from this data directly. Now the question is why we cannot do, because this reductionist approach, it does not holds good for the biology. Why is that? Because as I said, the reductionist biologists, they used to think, or molecular biologists, they used to think that a biological function depends on a single gene or protein. But today we do know that a biological function depends on the interactions of the several hundreds or several thousands of the genes and proteins together. So here is just a glimpse of the idea of how the complexity of the biology. So at my left hand side, what you can see actually is a network of the computers, okay, which are connected through the internet of whole world in the year 2009. And my right hand side, what you can see is the picture of a simplified representation of a biomolecular network of a bacteria. Just if you look at the topology of this system, you can see that the level of the complexity is almost same. But on the top of it, these biological interactions are nonlinear. There's a lot of feedbacks. And what is noise? There's a lot of noise. What does that mean? That at this moment there is no mathematics available to decipher this kind of basically the complexity, what is a simplified, even a simplified representation of biomolecular bacteria network in the bacteria. And that looks like a dead end to understand the biology from a very design level of the principle. However, human race never stopped at the dead end. So they try to find a new way that how it can be go around. And from the end of the last century, people started understanding that this particular biological or some of the biological network has some similarity with the other network related field, like electrical engineering networks or the computer science or the basically the electronics. So those kind of network has a similar level of mathematical abstraction with the biological networks. The biological networks are extremely complex than any electrical network what we have know that. But however, this gives a simple way out, that is a complementary way to understand the biological systems from a systems perspective, from a networks perspective, and that's sometimes called as the molecular systems biology. Now, if this philosophy is correct, then it is also possible that we could actually generate small biological network following the electrical engineering principle, put it inside the cell and develop a new function. So basically you start from your protein and genes, you develop a biochemical reactions, but you are taking the rules not from the evolutionary principle, because which you do not know, but you are trying to take the rules from this, how the gate makes a modules. That electrical or electronics engineering principle you want to put in the biology and try to develop some of the new function. So this gives an way in the biology as a design-based or biological engineering as a design-based subject. So probably you have seen this movie, or probably you not. I'm not recommending to watch this movie, but what I like to tell you, so there is a very interesting connection between the Amir Khan in this movie Ghazni and bacteria. Now, what is that? So if you either you remember or not, it doesn't matter. So there was a scene in this movie Ghazni, and okay, so one of the problem with the Amir Khan, he actually forgets everything because he had some wound on his head, and he forgets everything at every moment. So at one scene, what you can see, this Ghazni actually chasing a guy. He's chasing. Sorry, I mean Amir Khan is chasing a guy, chasing, and suddenly at a certain road, there is a truck came and actually hit that particular guy. And exactly at that moment, Amir Khan looked completely perfect because he actually forgets everything what he was doing. Now, what I'm trying to tell you that what does this scene actually tells us about the memory is that if you want to do a next step, like when you're running, so you're making your next steps, you remember your previous steps because you are chasing someone. Unless if you can remember that, you looks perplexed. And exactly what happened when this Amir Khan forgets everything, he doesn't know that what he was doing. Now you come to the bacteria. So if you have studied, I mean, definitely you have studied in your high school biology, there's something called chemotaxis in the bacteria, which basically means if you give a food or a chemical, and then bacteria actually swims towards it, and that's called the chemotaxis. Now this bacteria has a two different kind of motion. The one motion is going towards and directly going straight towards the towards that food or chemicals, and another motion is something called a spinning motion. So it actually instead of going anyway, any side, it actually start actually spinning by itself. So what happens now? Think about a thought experiment. Now you have a let's say you have a beaker, and in the one side of the beaker you actually gave a cube of the sugar, and this sugar will diffuse from one side to another. So you get a high concentration near the sugar, and the sugar concentration is diffusing low to the other end of the beaker. And other end of the beaker, you actually placed some of the bacteria. And now this bacteria has sensor which can sense these sugar particles, and they start moving towards it. Now remember, the bacteria is only a micron size object. So if there is a Brownian motion in the water, that Brownian motion hits the bacteria, and it can actually change its direction to other direction. Now, when this bacteria is going towards the sugar, okay, it is going directly to the sugar by swimming. But if water hits and sent to the other direction, what this bacteria does, it don't go. Because if it goes, it has to go to the opposite direction. It stops there and starts spinning. So another Brownian motion, some turbulence of the water hits back the bacteria towards the sugar, and then this bacteria starts going towards that. Now, what does it mean? So this exactly means that the Gaussian means that if it finds that in a certain direction there is a huge the concentration of the chemical or concentration of the sugar is increasing, and he's going there, and you take it back to the other way, then he can actually compare that in this way there is the sugar concentration is decreasing, it is less. That was he stops. But to stop it, it has to remember the previous instance, right? Otherwise it cannot stop, because but it can remember. Now believe that bacteria doesn't have brain. But how this bacteria transiently remembers the previous instances, right? So that is the question, and that question actually answered through what I call the first example of the synthetic biology. So I'm not going to the biology here, but what they actually developed, they gave a artificial system inside the bacteria where you can actually give something called an IPTG, which is a very small molecule chemicals. And this small molecule, due to the small molecule chemicals, there is an increase in a protein which is called green fluorescence protein. And if you take that IPTG off, still it is producing. If you give another chemical, ATC is coming down. I'm not going to the biology, but what I'm trying to say, what they actually developed a simple memory switch. It can remember this IPTG has been given. That is why when IPTG has been taken off, it still stays on. It's exactly like a switch. So as an engineering student, you do know is RS latch. So to make this kind of the system, what this is a simple memory system what they developed in the biology, and they took the engineering principle from the switch. Now, what is the biological manifestation of a RS latch? In qualitatively, what you need is two mutually repressing promoter gene pairs. Whatever that means, you don't have to go in the details. But what I'm trying to say, you have a qualitative principle, but just knowing this qualitative principle, you cannot make this memory because it also has a quantitative principle. This quantitative principle says only at a certain parameter level, this system will work as a memory switch or as a bistable system. So if you have studied nonlinear dynamics, you have probably studied this bistable or monostable system. So any other parameter level, you always get a monostable system, not the bistable. So when you know the parameters, now whatever your genetic system you're developing, you can actually change its lot of reaction rates like transcription, translation, their interactions with the DNA, so on and so on, and bring those parameters in the level of this bistable regime so that you can make this the memory switch. So this I can tell you, or you can understand, that it's completely it is very different than what is the conventional biotechnology or the genetic engineering we know today. Because here, first you need a design. Similarly, when you are developing a design in mechanical engineering, when developing something in electrical engineering, and so on, right? So that's in the design. So that's the difference between the biotechnology and the synthetic biology. Now, I'll give you one example. And after like 10 years of that, or 12 years after that, people actually develop. I mean, it came as something like biological computers destroy cancer cell. So what actually they developed a classifier circuit. So any students of electronic computer and electrical engineering, they know that what is the classifier circuit is. So this classifier circuit and actually can sense a set of microRNA inside the bacterial cells, right? and there's a in cancer cells, and in this cancer cell there are 6 microRNA which has a particular type of the expression. Some of them actually express high, some of them actually express low. So when you put this your biological classifier circuits inside the cell, if it match with those microRNA profile, it produce a protein and kill the cancer cell. But if it is not, if it doesn't match, it is not going to work, this classifier circuit. So you can see again in this example, I can show you that how you need a design first and then make some biological function similar to the other engineering. Now, this is another example, and a very interesting example here, because people developed a bomb detecting plant. So what does that mean? So if there's a bomb is coming, if there's a bomb nearby, and this bomb is always giving you diffuse some of the chemical molecules, like explosive molecules. So this plant, engineered plant, can actually sense that, and then the green plant turn into white. And that's exactly what I'm trying to show that here. So when in general it is a green, and at the end you can see it is completely white. So it can actually sense a TNT or RDX, and also it is a machine readable. You can see in the machine the colors how they're different. So this is, I just gave 3 different examples of the synthetic biology. If you want to do that, a big part I actually skip, and that big part is the how is it actually affect. I think the 80% of the chemical engineering of the future will be affected by the synthetic biology. But as your institute doesn't have a chemical engineering field, I'm not going into the detail, but I'm going something else. So this is a very, very pathetic news. So this guy in 2005, 2015 it came, and his name is Alankrit. I know this guy personally because he was 1 year senior to my son's class. So when my son is 4 years old, probably he was in nursery or KG, and he's just 1 year ahead of him. So what he had a problem, this guy has basically his heart has a congenital defect. So what he needs a heart transplantation. So what they got at the end, they got a heart from probably from Tamil Nadu, where a 26 years old guy died from a road accident. So got that heart. But the problem is, this Ankit is just a 5 years old kid, and the 26 years old heart cannot actually fit in here, and as a result this guy actually died. Now I come to this moment that this kind of the system, I mean this kind of so many fatality and so many pathetic news can only be avoided if we can able to make a whole heart from the patient's cell in vitro, that is meant outside the body. Then we can actually make a perfect size height for Alankrit and actually put that. Now I would like to emphasize here, this is the situation where if you bring all the doctors of the world together, they cannot do anything, right? If you bring all the resources of the earth together, all the money together, you cannot give life to Alankrit. So this is exactly the era, exactly the responsibility of the scientists, engineers, and the technologists to develop something heart outside the body. Otherwise, this kind of so many Alankrits in our world every day will die. So this is things that what the responsibility of a technologist or engineer or a scientist in this here. So people try to develop this, right? But this is a very difficult thing. So at this moment, what the people actually they do, they use something called the material scaffold. The material scaffold can be made through the 3D printing. I'm sure that as an engineer, you all know about the 3D printing. And that on 3D printing material, they actually put some of the cells. So the cells takes the structure of that organ. So this is a pig's urinary bladder that people actually make outside the body, right? And people try to make a different kind of hard valve, developing different kind of materials, programming materials, giving some scaffold, put the cells and try to do that. But we cannot make a heart at this moment. So our technology is not there. So this is one technology people are looking for in the tissue engineering, developing some materials with scaffold. Now, I'll just go and check that what could be the other way we can take from the science and engineering, which may help in one day to develop a heart. So this is a particular, I mean, if you have studied, I mean, probably you haven't studied this a particular kind of reaction, and it's called a BZ reaction. Okay, and what is that? It needs only 4 different chemicals. You mix them together, okay, and stir it. Okay, and now you just wait. It starts from a kind of reddish violet kind of color, and then you see after 10 seconds it goes to the blue, and again it is going towards the purple, and that violet color, this violet again goes to the blue, purple, and go. So you see this reaction is oscillating, right? So whatever in your chemistry study, whatever studied in your first year or second year or high school chemistry, you will always see there's a reaction A plus B that goes to C, like in acid-base titration, right? There's an endpoint. But here you see there's no endpoint. This reaction is oscillating. That's a very important reaction. I'll tell you that why in a moment. And then if the same reaction you do in a beaker on a petri dish, but this time you don't stir it, it is unstarred, you just keep there, and what you can see there's a very interesting patterns are emerging from this kind of the system. Now the question is, this is a very old chemistry, people died and people do not know what to do with it, right? And people stop there, and today we know that how important this kind of chemistry is. And so you probably know this person, right? Alan Turing is the father of the computer science. Now, what Alan Turing, you all know that developed this universal computing machine, that's what's called the Turing machine, and that is the fundamentals of the whole computer science. Now, and here I just wrote this apple. Do you know what is the connections between this apple and Alan Turing? Let me tell you. So Alan Turing is basically, I mean, he was a gay person, and that time in that era in UK, this kind of gayism was a criminal offense, right? So what the judge told Alan Turing, either he has to go for a death sentence or he can take a house arrest, and then they should allow the doctors to actually treat him. So because they used to think that gayism is basically a disease. So definitely, other than the death sentence, he chose the house arrest. But he was very depressed. At the very end, he actually committed suicide by injecting a poison inside an apple and then take a bite from that apple. So Steve Jobs gave respect to this father of the computer science by giving, naming his company as an apple and giving this figure a bite taken from by Alan Turing. Now, we know this Alan Turing is the computation, I mean, it's the father of the computer science, but he's basically a mathematician. So he developed some other mathematics to explain a certain kind of the pattern formation. So this is something called the developmental biology. What is developmental biology? Now, you remember that, you don't remember, but I'm trying to say that we know that all of our life started from a single cell. When I was in, or you were in your mother's womb, you started your life from a single cell. That single cell now makes trillions of the cells in our body, and many of the cells are very different. For example, our skin cell is different than the liver cell, our kidney cell is different than our eye cell, and so much. But very interestingly, the software of the cell, that is the genome of the cell, is the same information as in every cell. Either it's a kidney or the skin, the all same genetic information are the same genome. There is no difference in that genome. However, it functions very, very differently. So which basically means from a single cell, when you're starting the life, we are developing a different kind of the pattern. So some of them are going towards a kidney cell, someone is going to make the hand, some of the heart, and so on. So there's a patterning happening. So people do know today that this is a Drosophila embryo. Drosophila is like a fly, nothing else. So if you actually see that gene, this first, when this Drosophila embryo differentiating at the different organs, you can see actually the genes first patterning like that. And what this Alan Turing actually developed a theory, mathematical theory, is called the reaction diffusion system. If you take the chemical reaction and connect with the diffusion and nonlinear dynamics, you can actually explain such kind of the patterning. So probably the reaction diffusion system is the part of making heart or some of the or the whole developmental biology. So taking that approach, the synthetic biology, people actually develop a lot of patterning. So if you see this two pattern, colored pattern, this is just bacterial cells, not even the human cells or a Drosophila cells, just the bacteria. So you program the bacteria in such a way, when they grow, they follow this kind of the pattern. Now, here, one of them is a computer simulation, one of them is an experimental behavior. Now, just looking at it, you can see it is very difficult to distinguish that which is in the computer simulation and which is an actual experiment. So you can bring with the help of the synthetic biology, it is exactly coming to like the way simulation you design a car, the way in the simulation you design a rocket, and suddenly you can design the biology in almost at that level. So this is the kind of the patterning of this, right? So people actually increase this number of patterning. Here you can see 1, 2, 3, 4 patterns. Here I think there are 7 or 8 patterns. You can actually develop those kind of patterns, and all these patterns are mathematically predictive from the beginning. So this gives a very good idea to us that what we can do in terms of making heart. So you can start going towards that level, definitely far behind, maybe, I don't know how much it will take, 25 years or 50 years to make a heart. But this is, again, people try to, because all these things in bacteria, what I have seen, that is happening in the plate. But actually, heart is a 3-dimensional object. So here, people actually develop a 3D pattern. This 3D pattern is not with the material scaffold, before I say, is in the tissue engineering. He had just the cell. You give this program inside the cell. So cells are making this 3-dimensional pattern with 2 different genes, red and green, are expressing at various level. So this is very exciting at this moment. Now, I'd say this kind of tissue engineering is much more easy if you do in space. So in space, what is the one characteristics, and that is that absence of the gravity. Definitely, that's not an absence of the gravity. It's a microgravity. So that gravity of the Earth and the gravity of the space has a huge difference in terms of its magnitude, and that's called the microgravity. So here, people, as there is no gravity, you can actually develop a 3-dimensional object using the cell in space. So in the two of these things has been done in International Space Station. So this is a liver tissue they actually developed, grow in the space, and here they produced a colon cancer tumor on the space just by giving the cells. The cells actually grow in the 3-dimensional pattern. So this is very exciting, because probably this gives us an idea that the microgravity can assist making those kind of the heart of the 3-dimensional object when you actually connect it with the synthetic biology designed to itself. But space is not stops here. Space is very, I would say, is a frontiers of our future. And what, as an engineering student, you may or may not know that you can actually do semiconductor processing in space, and that would be advantageous in many, many different ways. So to make a semiconductor, what you do need is a silicon wafer, right? Now, you probably know the silicon is extremely, extremely brittle. So when you make the silicon wafer in the art, you cannot make as a very big silicon wafer. Today, I think the capacity is somewhere around 8 inch. You cannot make more than 8-inch diameter of a single silicon wafer, because if you make it, what happens due to the gravity, this silicon wafer actually breaks by itself. So it's so fragile. So that's actually one problem making the silicon wafer on the art. Another is, you do know that today when you actually do this micro semiconductor processing, when you make a chip, so now every day the chip is coming small, and small. So now if you print something in the chip, so let's say call it as a wire, so the distance between 2 wire at today's point in 2020, it is 21 nanometer. So 21 nanometer means, it's, if I can want to tell you how to imagine, so you imagine the diameter of your hair. Your diameter of your hair is somewhere around 25 to 50 micrometer. So you imagine that 21 nanometer. Hello, is there any question? I heard something.
No.
Okay. Yeah, sure. Yeah. So this 21 nanometer. So compared to your hair's diameter, you can see 21 nanometer is so small. So now Earth's atmosphere is full of dust, right? You have seen all the time, right? So many dust and very small particles. So any small particles, just more than 21 nanometer, if drops on your semiconductor, then the whole semiconductor is gone, because your wear is just the difference between the two wears is 20, the distance is, the gap is 21 nanometer. So what you need, this is an clean room. It's an Intel clean room, you can see here, and everybody is look like they're wearing the PPEs. Exactly, they wear the PPEs like an astronauts or these COVID doctors, and because you don't want any particle to actually come into your clean room. Now this is a very costly business, because to make a big room as a clean room, right? And now people actually show that you can do all these things in the space, and that would be very good, because in the space you don't have a gravity, so you can make a much bigger silicon wafer. At the same time, there's space, there is no dust, so all the clean room facility you can get in the space. The question is, what is the cost? Now, very interesting, I can tell you that I think it's in 2015 or 2017 data, I could not remember. So there is one cost estimation was done, and this cost estimation say that taking your all silicon wafer, all the silicon from the earth, making those silicon wafer, making those chips, and bringing back to the earth, it takes around 20% less cost than making the whole thing on the earth. So you can see that the space is in a new frontiers to do this, a lot of this technology. But where is the challenge for this space? I think the main challenge comes here. So let's say, what is the next space travel we're thinking of? And definitely the human think about the space travel is going to the Mars. Now, going to the Mars, it's not as easy to going to the moon, because it's, I mean, going easy. But what is the main problem? Because if you want to go to the Mars, you need somewhere between 1 to 2 years time. So coming back is another 1 to 2 years. And then you want to make a research based on the Mars. You just want to go and touch the soil of the Mars and come back. That's no. So if you have seen the movie Martian, exactly that's like that, right? So here, the problem is that if you go for this 4 to 5 years on the space, going to the Mars and coming back, so the resources in terms of the materials and the terms of the food, you cannot take it from the earth. It's a huge amount of resources. We don't have that thruster to send all these things to the space. So there's a huge amount of the payload. So for example, if you want to have some biryani, you cannot take it from the arsenal on the Siraj. You have to cook on the Mars, but you don't need only to cook on the Mars, but you have to grow all these ingredients, starting from your meat, starting from your rice, starting from your joyful jayatri, whatever. You have to make using the Mars resources on the Mars. Then you can actually have the biryani on the Mars, which basically means how we are going to do that. So people do believe, you can see this NASA poster, they believe the synthetic biology is the key for the future space travel. Because instead of taking a lot of payload, what you're taking only few engineered organism and bring just few vials, and you bring it to the Mars, and this organism using whatever the Mars resources, they will make your road, they will make your radiation resistance habitant, your materials, your energy, and everything, food, your drugs, and everything. So that is one way that synthetic biology is the key to the space travel. Now, so this is, I try to tell that this is another way that synthetic biology can change the future. And another way. So here, you probably heard about the robotics. I mean, probably many of you have a lot of, you probably also who are the 4th year or 3rd year undergraduate students, probably also make robots, right? So here is a very interesting robot, and that is called Spot Mini. It's a dog robot. It's an office robot. But you all know that if you want to make a robot, there are a few things you have to remember. There's a lot of sensors has to be there, right? And with the sensors, there's a lot of decision-making circuits you have to there. If you have a camera on it, it has to take the decisions based on this. And then you need energy. So you need a battery in the system. And what happens when you probably heard about something called the micro robotics, but if you go to the micro robotics, no micro robotics in the micron side. Why? Because of that. Because you need a lot of circuits starting from the sensor, the actuators, the decision-making circuits, as well as the battery. And you cannot bring a battery in the micron size level and all these things. So what we call is the micro robotics, it is basically a few centimeters big robot. Now, you think about bacteria, which is basically really a a micron size object, because this bacterial length is 2 to 5 micrometer, and on the in this way this is just 1 to 2 micrometer. So it is a real micron size object. You do not need a battery to put it here, because it can take the energy from the environment when you give the food. So the whole idea, if you can actually give all the circuits, that sensor circuits and decision making circuits inside the bacteria, you can do several kinds of work as a soft robotics or a micro robotics. So this could be a true micro robotics. And then people actually show this, people show in the last year that what they can do. So this structure is called a micro swimmer. People actually developed a micro swimmer. You can you design the micro swimmer based on the material property and the fluid dynamics of the liquid, and so that how easily or how controllable the swimming of these micro robots are. But there is no sensor, there is nothing. So now you want to put some energy in these micro swimmers on the itself. So what the people develop, they use a muscle cell connected with this micro swimmer. So it is a biohybrid robot, and this muscle cells, what this does, it can contract itself. When it's contract and then expand, contract, expand. This contract, expand actually give this whole micro swimmers to go at a certain direction. So now we can see that how cell material hybrid robot at the micron level can actually work as a very, can give something different. There is another example I actually forgot to give. I thought I'll give it, but let me tell you that very recently people develop a big biohybrid robots, where your robotic arm has some bacteria. This bacteria can sense chemicals. So based on that, that robotic arm can work in a liquid. So if they actually sense certain amount of the liquid which is sensed by the bacteria, this bacteria gives that output to the robot, and that robotic arm can actually, based on that, it decides what to do with that the liquid. Sorry, I forget to give you this, and this is also published this year only. Now I'll come to the change gear and come to the other things. Probably you heard about it. So this is a Deep Blue. It is a chess computer, and this is made by the IBM. And probably you do know, in 1997, this Deep Blue has been actually defeated the Garry Kasparov in chess, right? So this is a very interesting juncture where you are coming towards something called the artificial intelligence. We all know that, but the artificial intelligence, the way we define, I mean, there is a very much different kind of scales, very much different kind of the layers. But just take the artificial intelligence in very diffuse sense, that whatever we know by ourselves as the artificial intelligence. But this is something computer is playing a chess and actually beating a person, right? Today, I knew, you know about that. They actually even study the Go and play the Go, and they can defeat. So now, so what the people believe, that if you can understand a little of the brain, because the brain, whatever we are doing today, when I'm talking, when we're listening, or whatever we're doing, this brain is basically doing everything. So we have a billions or trillions of the neurons, have a trillions of the connections, and it can do all our intelligence, all our consciousness, and everything. And we do not know anything about the brain. What we know about the brain, some of the molecular principles, very little of them, okay? But we do not know how this brain function, how the brain got intelligent, how we have a consciousness on it. So people think that it's possible if you can actually simulate, or actually make in a circuit all the neural connection on a chip. And that is the, probably you heard, and that is the Blue Brain project. And this Brain project is few institute in the Sweden, and IBM are doing that. So at the current stage, what they actually developed, they developed a simulating a chip. They actually do all the connections of a rat's brain. Okay, now interestingly, they just make it. It doesn't work. It doesn't show any brain-like properties on that chip. Okay, so that is but this kind of failure is always happen because we do not know anything about the brain, and how to know. But what we know about the brain, that is also very interesting. Now, if you want to make this Blue Brain project a human brain like that, how much energy do you need? You need megawatt. So 10 to the power 6 watt, and it is the same as a mini hydropower. So it means that you want to make a single human brain kind of things, even if you don't know it is going to work or not, but you need this amount of the energy. But how much energy our brain takes? It's just 20 watt. Imagine the difference, 10 to the power 6 watt to the 20 watt. 20 watts in actual working brain, 10 to the power 6 watt may not be in a working brain. So what the biology tells us or teaches us that they use the energy of the thermodynamics in such a efficient way, and through that we actually started a new era or engineering, and it is called the ultra low power electronics. So there's definitely low power of electronics, and here is the ultra low power. Why do you need this ultra low power electronics? And specifically, people want to make those ultra low power electronics following the biological principle to make an AI processor. Why do you need that? Because of this. If you want to make a robot, bad or good robot or what is, right? You cannot put a mini hydropower on the top of the brain of this robot. You have to make a human-sized brain or a little more, right? So if you cannot make this processor or things is so energy efficient, you would never able to make a robot something like that who can actually do some of the intelligent work, and that is a very important part, right? So that's the start of that's how biology teaches to do that. So if you don't understand the biology at that energy level, you cannot make it. Now let me tell you one point. I don't have the slide for that, but again you tell you. So in a cell, if you take a single cell, right, and there are 30,000 reactions that are happening every minute. 30,000 different reactions are happening every minute, and they're happening in this very robust way by themselves. Now, if you take a simple operation on to off, this on to off you want to make in the single cell, and that on to off takes in terms of the energy 23 kT. But K is the Boltzmann constant, and you know those the Boltzmann constant is minus 34 or something. So this is a very, very low amount of the energy. Now, if the same on and off you want to do is in the engineering is not there, but even theoretically, if you calculate, let's say after 50 years, you want to make a nano switch which can take a very, very little amount of the energy to make an on to off operation, and it would take, right, 1,000 times more energy than this 23 kT. So in there's a theoretical limit of a nano machine, or when you use the inorganic materials to make that kind. We don't know why. So that's why it is very important to understand that how biology makes in that so tiny energy level. Now, let us go in the next, next 5 minutes or 6 minutes. I'd just like to talk that what our lab at Saha Institute of Nuclear Physics, what we are trying to do. One of the goals of our lab is can we make bacteria artificially intelligent, right? So, I mean, don't think that bacteria as an artificially intelligent means it will like work as a human or work as a big robot or a Terminator or something. But some important, interesting work if this bacteria can be done if you make them artificially intelligent. So to do that, we look at this artificial neural network. So you do know that, as I told you, that we do not know much. I mean, probably 99% of the how brain works. We know 1% in very detail, but that's probably 1%. So this from 1%, if you take a mathematical abstraction, so that's if you have a neuron and you have the input x1, x2, xn, which are coming from the other neuron, or it is coming from the environment, they combine it linearly, and then they produce through a transfer through a nonlinear function and give its output y1, ym, and this is connected to another neuron and so on. So if you want to do that in a paper and pencil, you want to make a artificial neuron, what does it mean? That in this neuron, whatever you just drawing it in your paper and pencil, there are x1, x2 to xn, different kinds of signals are there, and this x1. So first, all the signals are got added up. It's called a summer. This summer is exactly a linear combination of all this input along its weight, which basically means that x1 w1 plus x2 w2 plus x3 w3. Where this w as the weight, which actually say what is the fraction of this or what is the weightage of these inputs is connected. The first thing is the linear combination. Then this linear combination function is went through a threshold unit, or sometimes it's called activation function. Generally, it is non-linear. and you gives an output. So you got an output linear, non-linear output from this kind of input. And then if you just connect a lot of artificial neurons by like this, then what you make something called the artificial neural network. Though this artificial neural network is completely very simplistic version of a brain. Even probably we don't know if the brain works in this way or not. But even if we use that, it can give some very useful tasks. For example, probably all of you use Facebooks, right? And that Facebook, if you have when you upload a photo from a picnic or from a party or something, then what you see that the Facebook actually asks you, hey, would you like to tag this friend? So how this Facebook actually find or identify a face of your friend from a billions or millions of different photographs. And that is the answer is the artificial neural network. In the Facebook server, generally the artificial neural network they use, it probably it has somewhere about 50,000 different artificial neurons in that artificial neural networks. Now, in the most of the cases, again, 98% of the cases, this artificial neural network is implemented to softwares, those are the computers, right? But as I said, if you want to make a real robotics, you cannot just connect a remote server to a robot. That's not a good idea. So if you want to make a robot intelligent, you have to actually do a hardware implementation of the ANN, not a software implementation. So that's why there is one research field, and this is who developed a lot of different kind of neuromorphic chip, so that the whole ANN can be performed in the chip only. So then put in the chip. So that is basically a lot of inorganic material based chip people develop. People actually do that ANN through the photonics. And people showed that it can be done in the in vitro DNA computation. Means they can in a test tube giving just the DNA into it. What is my lab is interested, or the question we do ask, that can you make a bacteria as an artificial neuron, and then a population of the bacteria can work as an artificial neural network which is made by this bacteria. And to do that, we chose something, a decoder encoder. I'm sure that most of the engineering students, you know what is the decoder encoder, but who doesn't know, just to give a brief introduction. So this decoder encoder are always used in a lot of wireless communication system. For example, if there's a walkie-talkie, and this walkie-talkie, when I'm talking, then this sound signal goes to the electrical signal. That electrical signal is first got encoded through a circuit or device called encoder, and that encoded to a fewer bits, and that information is sent to the other instrument, and other receiver actually receive that, and that's encoded signals actually first decoded through a decoder, and then actually come to as a sound signal, and you can hear that. So there's a lot of communication system in the avionics industry and so many things. What I'm just trying to say, that the decoder encoder is a very important system, and what we would like to do this decoder and implement decoder and encoder using bacterial artificial neuron. So I'm just trying to give you showing the result. So this is a 2 to 4 decoder. This is the truth table, and this is the X and Y at the input. And based on the combination of the input, it has 4 different outputs. And if you can see here that diagonally there is 1, 1, 1, everywhere is a 0. So what we make this system using bacteria, but not following this electronic engineering principle, but just following this artificial neural network way. And I'm not going how we're doing that, but there's a lot of molecular engineering is there. And then we feed these equations, a lot of mathematics to change that, how this weight and bias is going to be adjusted, and so on, so on. And finally, when you actually make this network, and this is an experimental result, you can see diagonally there is only fluorescence like red, purple, green, yellow, and everywhere it is black. So exactly it is like that. Diagonal is 1, 1, 1, everywhere it is 0. So we're excited because you can make this 2 to 4 decoder with the bacteria using ANN. We have done encoder again. I'm not going. This is a truth table. You can see this is much more complex than the decoder. So this is our ANN with our artificial bacterial neurons. Because remember, this is in hardware, because it is actually happening. It's not a software. It's not the mathematics. This mathematics is actually happening with the bacteria. And then when you do this kind of experiment, you get the results. And if you match them, they are actually the same. So we also try to do something, right? You probably heard, I don't know about this, anything about the quantum computer. Okay, you may know much more than me, but just what you say that in quantum computing, there's the platform of the, if you want to do through logic gate kind of a base platform, then the irreversible computing, what we do in AND gate, NOR gate, NAND gate, it is not the way. So you have to do through reversible computing. And this reversible computing has two different aspect. One is thermodynamic reversibility, another is logical reversibility. Now, what you have studied in the thermodynamics in your chemistry or the physics class, specifically in your first or second years, that thermodynamics, specifically the reversible thermodynamics, that is not applied to the biology, right? So when I said we have a reaction, A plus B goes to C and it ends, right? But in the biology, the reaction doesn't end. This is, I'm talking, you are listening, or you're pretending listening, probably you are, I don't know, sleeping, okay? Or whatever you're doing. So all these out of the equilibrium, right? You cannot do, the reaction doesn't stop in the biology. If the reaction stops, means I dead, right? I'm dead. But we are not dead. So we are out of the equilibrium. So whatever the thermodynamics what has been used in a system is basically irreversible thermodynamics. And people use this thermodynamics in basically in the chemical engineering, in engineering field, and the whole biology is irreversible thermodynamics. So however, therefore, we cannot attain thermodynamic reversibility if you want to make through this bacteria ANN. But what we can attain, what we call is a logical reversibility. And let's see what is the logical reversibility is. Now here, main thing of the logical reversibility, that it has the information get preserved. So the input looks exactly like the output. So this is a Feynman gate, or Richard Feynman actually proposed this gate, and it is also sometimes called controlled NOT gate. So now if you see, if it was a kind of an AND gate, you will see only when there is a 1, 1, 1, then your output is 1. Everywhere it is 0. So information is not preserved for the other three cases. But here you can see that A and B, if it is 0, 0, P and Q output is also 0, 0. If it is 0, 1, it is 0, 1. If it is 1, 0, it looks here 1, 1. Definitely the output is for this particular, it is not preserved in straight way, but in a zigzag way it is preserved. So this 1, 0 is come here, 1, 0, and this 1, 1 is come 1, 1 here. So you can see that information is preserved. So what with these things, we actually developed this Feynman gate with this bacteria ANN, and basically what you can find, this is the actual result here. Okay. So another area of our work, what we do, which is called the space synthetic biology. As I say, the synthetic biology could be the key for the future space travel. So Adam Merkin and their group actually proposed there are 6 areas of the future space technology where synthetic biology can have the contribution, and starting from resource utilization, manufacturing, life support, space medicine, space cybernetics, and terraforming. So in all of that, what is basically we need in some of the cases, the microgravity is an integral part of the system. So this microgravity, what does, like when there's an astronaut goes to the space, that immune system got deregulated, as people know from the Apollo mission. At the very same time, our own gut bacteria, which is in our gut at this moment and doing nothing, no harm to us on the Earth, if we go to the space, our own gut bacteria got virulent. So this is a very clear combination. But people do not know that how microgravity affects these tiny particles or tiny bacteria or tiny cells to make such kind of the phenotype. People studied it very well in space and microgravity simulator on the Earth. However, somehow their statistical analysis technique is not that good. You cannot take a lot of information out there. So what we actually developed a systems biology pipeline using several statistical tools and machine learning algorithms, and we got several new insights what other people could not find from their own data, right? And then we published that. I'm not going into the detail. Just one thing I want to mention that here we used something, a very different engineering, and we use something called non-negative matrix factorization. It is a machine learning algorithm, and specifically people use for the face recognition. The same machine learning algorithm we have used to actually analyze the space-related genome data. So this is interesting, right? How this engineering and biology are doing very well together. And when we actually publish those papers, this just we got featured in Times of India, Nature India, some other newspapers, and so on. Okay. So whatever we got insight from there, our goal is to develop something called a microgravity sensing biochemical device. So we developed a synthetic gene circuits, and we actually tested in our microgravity simulator, which we have our lab, which is designed by the NASA, and you can see the result. When there is a microgravity, the signal is almost zero. When it's gravity, the signal is around 30. So there's almost 30 times difference in the signal. So we're happy that we can do that. And with the microgravity, we can control the cell shape and cell division process. Here is so in Earth's gravity, so we engineer the cell in such a way, in the Earth's gravity, you see the cells looks like some elongated way, right? Elongated, this is not the exactly cells thing. So now our device say that when you actually put in the microgravity, this shapes has been cured, and now you won't see this kind of elongated shapes. So we're very happy that we are able to make world's first microgravity biosensor, and we can use this. We can use this to actually control many cellular processes with that. Now, this you can think about an engineering problem, right? Now think about the energy. Now we do need a lot of energy when you actually have the street lights. Now think about that amount of the light takes a lot of amount of the energy and lot of the cost. What about you can actually develop this kind of the solution to the energy? This is taken from the movie Avatar. So if you make the tree, whatever's happened just in the roadside tree, they glow like this, then you do not need those kind of electrical things. So this is another way to think about the solution, engineering solution for a lot of this problem. And with that, I like to stop here. And this is my small group. They're all my PhD students, and he's the scientific assistant. And this is the funding from where we got to do our research. And thank you very much for listening.
Thank you, sir. Thank you very much. It was indeed motivating, enlightening, informative, and very interesting. I think the session is now open for questions, sir.
Yes, please.
Okay, so I request all the participants to please come forward with their questions. They can also write it in the chat box and I'll read it out for them. Do the participants have any questions? So please, you can come forward with your questions.
Student can put their questions on chat box also.
Yeah, I request them to write their questions in the chat box, but still in the chat box I don't see any question.
Sir, one thing I'd like to ask.
Yeah.
Hello.
Yes, yes.
So, how synthetic biology helps in today's biomedical instrumentation?
Biomedical. So see the okay. So the biomedical instrumentation. I mean, one of the important aspect of the biomedical instrumentation is basically the robotics and the AI. And I would show that with the synthetic biology, what you can develop a lot of biohybrid robots. So, for example, so let's say inside the human body, you want to see something, and now if you have an engineered organism like the bacteria, you put through your bloodstream. Through this bloodstream, this bacteria can go at a very different spaces, and then actually, if you have a disease state, like if there is a cancer is growing at some point, there is the microenvironment signature of that cancer. It will produce some chemicals, so that if this bacteria can actually identify that. So and this bacteria it can be full of your drugs inside it, so it can actually deliver those drugs there. So now instead of that, if you use a biohybrid, not just a bacteria, you can micro swimmers which is connected with the bacteria, so you can actually using a magnetic control, you can actually control the movement of this micro swimmers. But the bacteria will take all the biological, it actually take the biological information from it, and it will send to you through your whatever the micro swimmer. So this is the one area to actually go for the biomedical instrumentation. So the other area, I mean, the whole diagnostic platform is basically people are trying to put everything as a bacterial sensor. So I said there's the bacteria, but it's not need not be the bacteria. People actually developed a lot of mammalian cells, and what you can put in your, for example, in your brain, right? And this brain, it is an optogenetically engineered, which basically means that you can actually control that the cell function by applying light from the outside, right? And those are the things you can actually do that. There may be other also. I don't know all of them, but this is just a few things.
Okay, thank you.
Yeah.
Thank you, sir. We have a question here from Polashnath Das. He is
Yes.
asking whether we can make solar panels using human hair.
Okay, so I really don't know that if it can be done. I mean, I never thought about in that range. But what I can give you information for people developed using plant material. You know, probably you do know that people use the plant material. Now, very recently means like few years back, they actually use some of the carbon nanotube antenna and put it on the plant's leaves. So now, when the plant leaf actually takes that energy from the sunlight, it has a limit. It takes like 10% or something. So when they put this nano antenna, so now through this nano antenna, a plant can take more than 30% of the solar energy into it. So this kind of the technology can be used as a solar panel because you're taking a lot of energy and putting inside it. So, but you said that human hair. I don't have the idea. That's why. But maybe, I mean, what is your I don't know. If maybe anything in science, what I can say in science and technology, anything is possible. Anything, right? Just we have to find a way. That's all.
Thank you, sir. We have another question from Jhornotsho Dashgupta. He says that if we are able to make a body part in space, how good will it be for the patients?
No. So the question is, what I'm telling, that doesn't mean that we have to make in the space. So what makes this body part? Because if you make the space, you have to bring it down. I mean, that people can do that. I mean, but what you need the microgravity. So as I showed that we have for example, we have a microgravity simulator in the lab. So there is a lot of other microgravity simulator. So we have a big microgravity simulator. So you put, instead of a simple reactor where you want to grow your cells, you put your microgravity simulating reactor, the whole thing. So the microgravity will help you to make this three-dimensional. You don't need to go to the space. You can do those kind of things on the Earth. But in terms of the semiconductor processing, no, because the dust is the problem, and you cannot make the semiconductor inside the water. So that case, you have to go to the space.
Is there anyone who wants to ask something else?
Hello.
I don't see any more questions in the chat box.
Hello, am I audible?
Yes, sir, you're
Yes.
audible.
Sir, one thing you just want to say that here so many students are there. So how they can make their career in the field of synthetic biology? So please give an outline.
Oh, sure. So first thing also I'll tell that the synthetic biology actually developed by the engineers only. Okay. So for example, my PhD supervisor, who was a space engineer, he did his PhD on the space engineer, and his algorithm for making the robotic communication has been used one of the rover Mars mission. So now when he was listening to a talk to synthetic biologists that's who actually developed the fast toggle switch and the memory switch, so my supervisor realized that what about the algorithm he used in basically in this communication of the robotic device, the same kind of the algorithm those guys are using in the connection of the genes. So he interested in synthetic biology, went to synthetic biology, and when he came as an assistant professor at University of Toronto, I joined with him. So basically my background is also in the after BSc I also did a BTech on chemical technologies, petrochemical engineering. So that is also so a lot of people actually in the synthetic biology came from this the engineering. So I think the one way to go, I mean one way, there's something called the iGEM. So if you just search in the Google, and it's called the iGEM, International Genetic Engineering Machine Competitions. So there is a lot of engineering college from the India is actually take participation on that. So if you are interested, you can actually go, and that competition give you is a lot of different learning resources, a lot of other resources. So this is one way to do that. Second way, so the synthetic biology is if you want to go, there's two ways to go. One is if you want to do some higher studies on it, so you can think about doing PhD on synthetic biology. And nowadays I think US doesn't need any GRE, right? So last couple of years they are not asking for the GREs. So if you have some synthetic, so first thing I will study a little bit on the Google search. If you can do the iGEM, that is good. If you can take some your summer, okay, instead of a company when you do your industrial research, I mean if you take some lab where people are doing synthetic biology research, you have a certain kind of experience, and then go out at very good. So I know like a couple of students definitely did from Sipur. They're specifically electronics, at least 2 electronics engineers actually worked with me during the summer project. So that is one, and then you can do your PhD. And another thing, there's a lot of synthetic biology company, they ask for because there is a lot of the data, they do need someone from the computer science or electrical or electronics who can actually analyze those data in various ways in the data science or something. So another aspect, if you can go such kind of a company, if you look at, because I know that you don't look the companies other than your hardcore or IT system, but these are the companies they always look for those kinds. So you can think about connect those with those companies. So iGEM is a very good resources. You go and you learn many things, so which are the companies, what they're doing there, and where to connect, how to do that. So this is a very useful resource, I believe.
So thank you. We have Konko Kundu asking where India stands in the field of synthetic biology.
Okay, so everything India starts for after a long time. So there's a so at this moment there are so okay, let's say how this synthetic biology evolved. So what I presented here, that the synthetic biology has started in that way. There is another part of the synthetic biology which is called synthetic metabolic engineering. Basically, you are producing a lot of chemicals inside the organism, bacteria, yeast, or so on, and these chemicals can be useful for many industrial processes. So in India, whatever the synthetic biology community is, there 90% of the community or 95% of the community are making those kind of the chemicals on those things. So those are in my opinion, they are not real synthetic biologists, because those are again is a genetic engineering. You are doing industrial microbiology. So from a electrical or computer science perspective, what the circuit things are the basically they're electrical, I mean synthetic biology, and there is only few groups are doing. Definitely my group, and there is a Sonak Shen, and he's in IIT Delhi. So he is trying to, I mean, he has an experience in both modeling and experiment, but he's just doing the modeling. He hasn't started the experimental things. And there's Amit Ghosh in Kharagpur IIT in Energy Science Division. He's doing trying to something like that. And there's a Professor Pabondhar in JNU. So there's a community is very less, but what I can say, the opportunity is enormous.
Thank you, sir, very much. We have Adrish Day asking, which is better, biotechnology or bioengineering?
I mean, first thing is that what do you mean by bioengineering, biotechnology, and better in what sense? So what I showed here is basically that how our future evolves. So I mean, you can say the synthetic biology is a part of the biotechnology or bioengineering, right? Because but the whole goal of the synthetic biology, you want to come with a design-based perspective. But in biotechnology, it's not a design-based. There's like mostly it is like a trial and error. And but in trial and error, you cannot do many things, right? That's I showed, like those kind of switch, those kind of artificial neural network, those kind of classifier for the cancer, okay, or biohybrid robotics. Those you cannot do with the conventional biotechnology. What you do need, those kind of the principles, design-based principle, and that's the synthetic biology. So I mean, there is nothing like to say that what is good or better in what sense. I mean, they're all same. It is question is what interests you the most. If it interests to making the chemicals, you can definitely go to the biotechnology. If it interests to do this kind of research, what I just showed you, then definitely the synthetic biology is the key.
Thank you, sir. Dharnotra is again asking that if you can go there if you have a mechanical background.
Yeah, exactly. Because there is a many people come from the civil, many people come from the mechanical. Because many of my when I was doing my postdoc and a couple of students who are doing the PhD in our lab at chemical engineering department, and they are basically the mechanical engineers. There's undergraduates from mechanical engineering. So one thing I'd like to tell you for all the students, that science or engineering or the technology, here we divided like electrical, like chemistry, like things. But when it comes to solve a problem, you cannot pretend you are something like that. So if I need to know the mathematics, I have to know the mathematics. If I want to learn the biology, we learn the biology. So we, specifically in India, people are, I mean, we are responsible, like, right? So what makes that we said, oh, you are electrical engineering, you're just electrical engineering. You are a physicist, you're just a physicist. It's not like that. It's never been some kind of thing. You start with one undergraduate degree and you can do anything you want. It doesn't matter if you're starting from the linguistics and you come to the physics, right? We know that's those kind of things are like de Broglie. De Broglie was a linguist, not a linguistics. De Broglie was like a music students basically, and he got the Nobel Prize in physics. So there's no bar. You can come from it. Is only depends on what you really want to do. If you really want to do from the heart, do that, right? It is not like how much your salary. I mean, if you think of the engineering, I mean, today's in Indian engineering system, I must say that we lost the charm of the engineering, because we want to think in terms of that what we can do in terms of the salary, what is the product and something. But engineering is not about the product. It's not about the economy. Engineering is something that something does not exist in the nature, and we want to make that. So that's the creativity, right? That's creativity is the engineering. So that is, if you have the passion of the engineering, you can do that. Then Elon Musk has something to do which is only the passionate person can do, right? You cannot just do like it. So it is all up to you. I believe that if you want to do something, you want to be a singer, you want to be a synthetic biologist, you want to be a physicist, you want to be electrical engineer, it's just up to you. Okay, so you can do anything. Sorry, I cannot hear anyone.
I had gone mute, sorry.
Yeah.
Thank you, sir, very much. Thank you for your valuable speech. Thank you for your valuable time. And I would now like to request the head of the Department of Basic Science and Humanities Department, Dr. Rajdeep Chakraborty, for delivering the vote of thanks. Sir, over to you.
Hello. Am I audible?
Yes.
Oh, thank you. A big thank you to you. Thank you for the wonderful session. Respected principal, dean academics, most valued invited guests, and my dear students. It's my privilege to propose a vote of thanks on this occasion. On behalf of the Departmental of Basic Science and Humanities, RERF, I extend a very hearty vote of thanks to the speaker, Dr. Sangram Bagh, for gracing your important work and sharing with us your findings and opinions today. Again, a big thank you to you, Dr. Bagh, for your effort towards today's presentation, and particularly the issues you have raised for a new emerging technology. So We are all inspired by your words. Again, I'm really happy to see the number of my beloved students who have participated in the program, and hope that the students can explore your thoughts in the future in the field of synthetic biology, which is shown by Dr. Bagh. Again, I would like to extend the hearty thanks to my colleague, Mr. Ovi Ghosh and Moinak Sarkar for the perfect logistic support and technical guidance that they have extended to us to make this program successful. I also extend my thanks to our principal, Dr. Muhammad Das, and Dean Academics, Dr. A.K. Shaw, for their support in various academic issues. But last but not the least, I am very fortunate to have a team of very motivated and dedicated colleagues in my department, without whom no events can become successful. Thank you. Thank you all.
And it's my pleasure to be here, and I thank you all for listening and inviting, and really I enjoyed. Thank you very much for this session.